When you hear the word lipid, you probably think of fats, right? Or maybe oils, but there's so much more than that. They are, quite literally, the architects that build the very boundaries that make life possible.
So let's start with a really big, fundamental question. How does life even contain itself? I mean, think about it. How does a cell draw a line in the sand, separating all its delicate, organized machinery from the complete chaos of the outside world? Well, the answer, it's all about the elegant and surprisingly simple structure of lipids. So here's our game plan. We're going to start with that big boundary problem, then look at the versatile building blocks that actually solve it. We'll see how they come together to form the cell's main barrier, the bilayer, and then, this is the really cool part, we'll explore how that simple barrier becomes a dynamic, living structure that's adapted for even the most extreme places on Earth.
All right, let's jump right in. Section 1. Creating the container for life itself.
It really is that simple. No lipids, no cells. End of story. These organic molecules are absolute masters at creating barriers. They build that stable, protected little bubble where all the incredibly complex chemistry of life can actually happen without being disrupted.
Okay, so if we want to understand the wall, we've got to start by understanding the bricks, right? So let's meet the fundamental, versatile building blocks of all this, the fatty acids.
Okay, look closely here because this difference in shape is everything. See the stearic acid? That's a saturated fatty acid. It's perfectly straight, like a ruler. But then look at oleic acid in the middle. It's unsaturated, and that double bond introduces a permanent kink into its chain. It seems like a tiny little change, but trust me, the consequences of that one bend are absolutely massive.
And here are those consequences in action. The straight, saturated chains, they can stack together perfectly, almost like a neat pile of firewood. That tight packing is what makes them more solid at room temperature. Think butter. But the kinked, unsaturated ones, there's just no way to pack them together tightly. It's like trying to stack a messy pile of crooked branches. All that extra space between them makes them more fluid. Think olive oil.
So with all these different shapes and kinks, chemists obviously needed a standardized way to talk about them, a shared language. Let's take this molecule, linoleic acid, as our example to see how they do it. The first method is called the delta system. It's super logical. You just start counting from the head of the molecule, that carboxyl group, and number the carbons all the way down the chain. So if you see something called cis delta 9, it just means the kink starts at the 9th carbon from the head. Simple as that.
But there's another system, and this is probably the one you've actually heard of, especially if you look at nutrition labels. It's the omega system. And all it does is flip the molecule around and start counting from the opposite end, from the methyl tail.
So when you hear omega-3 fatty acid, what does that actually mean? It just means the first double bond is located 3 carbons in from the tail end. That's it. It's the exact same molecule, just described from a different point of view. 2 different maps for the same territory.
All right, so we've got our bricks, the fatty acids. Now it's time to add some mortar and start building the larger structures, the ones that will actually become our membranes.
To do that, we need 2 key parts. We've got our fatty acid chains, of course, and then we have this guy, on the left, glycerol. Think of glycerol as the simple 3-carbon backbone that we're going to build everything on top of. It's our foundation. The assembly process itself is really elegant. The glycerol provides the platform. 3 fatty acids come in and line up. And then, through a neat little chemical reaction called condensation, they get locked on, forming these super strong connections called ester bonds. In the process, a water molecule gets kicked out. And voila, here's the final product, a triglyceride. Now this thing is an amazing molecule for storing energy. It's stable and it's completely hydrophobic. It hates water. But that's a problem for us, right? A cell membrane has to deal with water on the inside and the outside. A totally water-hating molecule just won't work. We need something with, well, a bit of a split personality.
And that brings us to the magic word for this whole topic, amphipathic. It's a fancy word, but the idea is simple. These membrane lipids have that split personality. They have a polar head that loves water, it's hydrophilic, and a nonpolar tail that hates water, it's hydrophobic. It's this love-hate relationship with water that forces them to automatically build barriers all by themselves.
So let's think about the geometry. What happens if your lipid molecule only has one of those water-hating tails? Well, when you throw a bunch of them in water, they do something really cool. They spontaneously form these little spheres called micelles. All the water-loving heads point out towards the water, while the single tails all huddle together on the inside, hiding.
And this is exactly how soap works, by the way. Those single-chain molecules are perfectly shaped to form these little micelles that trap grease. But here's the catch for biology. A micelle is tiny. You can't build a cell out of these.
To build a cell, life needs something way, way bigger. It needs this, a huge, two-layered sheet we call the lipid bilayer. So, what's the secret? What's the one tiny change in the design that lets you build this massive wall instead of just tiny little spheres?
You're gonna love this. The answer is so brilliantly simple. Two tails. That's it. Phospholipids, the main lipids in membranes, have two fatty acid tails. This changes their whole shape from a cone into more of a cylinder. And think about it. What happens when you stack a bunch of cylinders next to each other? You don't get a ball, you get a flat sheet. And that right there is the fundamental design secret behind every single cell membrane on this planet.
But, this basic sheet is just the starting point. It's the canvas. In our last section, let's look at how life adds specialist molecules to this canvas to make it work under all kinds of conditions, including some of the most extreme environments you can possibly imagine.
First up, meet cholesterol. And right away you can see this thing looks nothing like a phospholipid. It's got this bulky, rigid, four-ring structure. It's clearly a specialist, built for one very important job.
Cholesterol's job is basically to be a fluidity buffer. It wedges itself right in between the phospholipid tails. Now, if things get too hot, its bulky shape gets in the way and keeps the membrane from turning into a sloppy mess. And if things get too cold, it prevents the tails from packing together and freezing solid. It's like the Goldilocks molecule. It keeps the membrane's fluidity just right.
That's pretty clever, but nature can do even better. I mean, what kind of membrane do you have to build to survive in, say, boiling acid, or under the crushing pressure at the bottom of the ocean?
This is where we see some true molecular engineering genius. The membranes of archaea, these are some of the most ancient and toughest life forms on Earth, have three major upgrades. First, their chemical links are ether linkages, which are way stronger than our ester ones. Second, their tails are branched, which lets them interlock like Velcro to resist being torn apart. And third, they're built on a mirror image version of glycerol. You put those three changes together, and you get a membrane that is just unbelievably tough.
And that leaves us with a pretty wild final thought. We've seen how life has brilliantly engineered these lipid membranes to handle just about anything Earth can throw at them. So it makes you wonder, what new kinds of lipid architectures could we design? Or what might some other form of life on a world beyond our own come up with?